Thank you very much, and thank you very much for sticking it out here. We are close to the end, but thanks for hanging on to this. As you can tell from the title, I'm going to talk about adenovirus vectors, and the main thing I want to, and it's late, so the main thing I want you to get from this is that the platform I'm talking to you about has nothing to do with your grandfather's adeno. So, adeno's been around for a very long time, and I think that the platform that we now have has performance characteristics that meet what we're going to need for cell and gene therapy, as well as molecular vaccines and a whole host of other applications. Which one? There we go. Thanks. So a little bit about GenVec before I get started. GenVec is a pioneering gene therapy company, and we're focused on leveraging this proprietary adenovector technology to really develop a pipeline of cutting-edge therapeutics and vaccines. We have deep experience in adenovector development and proven capabilities in translating scientific concepts into clinical products. Our lead product is partnered with Novartis, and it's a regenerative medicine approach to regrow or to regenerate sensory hair cells in the inner ear. And this is a first-in-man clinical gene therapy, and no one's delivered drugs to the inner ear, let alone gene therapy. And our therapeutics and vaccines have been safely administered in over 3,000 clinical study subjects. And as I pointed out before, the technology platform we believe is suitable for a diverse range of applications, therapeutic approaches, and we have a strong IP position across the platform. So why should you even be thinking about an adenovirus vector, especially this late in the day? Proven ability to deliver payloads efficiently to cells, no incorporation in the cellular genome, can start thinking about in dividing cells hit and run sort of delivery and expression approach, where you can get into those cells and get out of those cells but deliver your payload. And we can also construct these with either brief or extended gene expression in differentiated cells. So why GenVec adenovectors? One of the main things that was a disadvantage to your grandpa's adenovector was pre-existing immunity. And the constructs that we have no or very low immune seroprevalence in human population. The second aspect is that we can delete multiple viral gene deletions within our vector. So these are all replication incompetent. And for this approach, it gives you a very large vector payload capacity. 12 kb is easy to be able to put back in. You can start thinking about multiple transgene expression cassettes within the same vector. And moreover, when you delete these viral gene regions, what you do is you reduce what happens to the cell as the vector goes into the cell. Vectors cause cell perturbance, can change the cell in a variety of ways, and by deleting these, it can be much more stealth getting into the cell. We also eliminate viral gene expression in transduced cells, which was another deficiency in first-generation adenos. We have proven manufacturing capabilities. Our complementing cell lines that grow everything that we will be talking about are on FDA master file, and our approach meets international standards of large pharma as well as regulatory agencies. So we think about using this technology across a broad spectrum of applications, and this is to depict some of those applications. And you can see the technology in the center therapeutic gene delivery up top, because that's what we've been doing with the technology the most over the years. That is our hearing loss drug. We're delivering a gene for regenerative medicine approach. They're also very useful for vaccines, in particular molecular vaccines, where we can encode an antigen and get a very strong antibody and T-cell response to the encoded antigen. And we've also begun working in cell therapy. We have a collaboration with Therabiologics, where we're using adenovector to modify their neural stem cells in order to provide an enhanced genetic modified cell. But we're also looking for approaches in all of these other areas. And we're a small biotech company. So how will we do this? What we'd like to do is to have a collaboration approach. And we're actively exploring opportunities to expand the platform into these applications, as well as others that you could think about. We want to be able to add complementary assets and technologies to our current portfolio, and we also want to maintain our own drug development, but also yours, and to be able to help in ways that we think that we can. I have an open innovation and collaboration approach, and we really want to unlock the potential of this technology for new medical treatments. So a little bit of detail on the vector platform. This is a portfolio of vector types that you can use. Some are human-based types of adenovirus, and this includes some rare human serotypes. And we found these to be very useful for sensorineural applications, in particular in the ear, as well as in the eye for delivery. And we have a suite of non-human adenovectors. And these come from both new and old world monkeys and gorilla sources. The new and old world monkeys I'll show you a little bit more data about. They seem to have great delivery performance in cells that are usually recalcitrant to normal adenovirus vectors. The gorilla adenovirus vectors have really great performance characteristics for molecular vaccines, as well as a couple of other things. We have the ability to genetically modify the surface proteins of the adenovirus vector to engineer specific cellular interactions, and we can build in and modify a promoter for specific cell expression of your gene, which actually allows you to start thinking about tuning the tropism of the vector to what you're looking for. You can also modify the transgene expression cassette and design for the level and durability of expression. And all of the vectors that I'm talking to you about grow to high productivity on our cell lines and are manufacturable and industrializable, all ready to go. This is a little bit of data, and it's a graph that looked at a baker's dozen of the new vectors, that is the B through N component on this scale, compared to Ad5 in a couple of different cell types. And the axis is the fold enhancement relative to Ad5. So you can start to see that there are vectors here that have 50 to 100-fold enhanced delivery to cells that Ad5 can have a tough time getting into. So there is an application for even cells that have classically been difficult to deliver genes to. This is our pipeline. The hearing loss drug is up at the top. There are a number of infectious disease approaches using molecular vaccine approaches, as well as our collaborations with Therabiologics on modifying neural stem cells. And in summary, safe delivery in humans, lots of patients and volunteers treated. Can get a large transgene capacity into these vectors. You're able to accommodate multiple transgene cassettes. This tunable tropism technology allows you to deliver to a broad spectrum of cells and tissues, and even selectively. There are a lot of serotypes with extremely low seroprevalence in the human population, which deals with some of the limitations that past technology has had using adeno. Proven manufacturability, very high productivity in our cell lines, and all of this meets international standards of both large pharma and regulatory agencies. And there's outstanding genetic and shelf life stability of these constructs. And we do believe it's applicable for gene therapy, cell therapy, regenerative medicine, and a number of other approaches that only your ideas will help us to explore this even further. I'm here at the meeting as well as Sebastian, and if you think of something and would like to talk to us, please do. We're available through the end of the week. Thank you very much.